Hi Ark, and hello to all the viewers. I am Sahil, and I work in the Department of Biochemistry and Molecular Biology at Tel Aviv University. I'm working here in the capacity of Post-doctoral Fellow in the lab of Professor Abdussalam Azem.
Wow.
And I'm very grateful to him for this opportunity. My work here is basically on deciphering the impact of genetic mutations on biochemical pathways and on protein steady-state levels.
Could you tell me more about your research here?
Yeah, sure. To let you know more about my research, let me give you a brief background. So we all know that the human body is made up of cells, and cells contain various organelles such as mitochondria. And mitochondria is also known as the powerhouse of the cell.
Yeah.
Right? And in humans, the mitochondria is known to have around 1400 proteins. But most of these proteins are not synthesized in the mitochondria. They are imported from the cytoplasm inside the mitochondria. And to facilitate this import, there are various complexes that are involved. And one of the complexes is TIM53 complex. Now, one of the subunits of this complex is TIM50 protein. And our work over here is to understand the biochemical and neurophysiological effects of the TIM50 mutations.
Interesting.
Yeah, so you can understand this by imagining that you need to enter the university by crossing a turnstile.
The one we have here.
Yeah, exactly.
Okay.
So if the turnstile itself is malfunctioning, it would be difficult for the students to enter the university.
Very true, yes.
Yeah. So similarly, these import complexes and the subunits of these import complexes, such as TIM50, if they are malfunctioning or if they are deficient, then we can expect that the mitochondrial protein import will be affected.
Yeah.
But surprisingly, in the case of humans and mammalian neurons, we found that this is not the case, as most of the mitochondrial proteins are still being imported despite dysfunctional TIM50.
Okay.
And it is very exciting for me to share my work with you, which is now available at bioarchives. And I would also like to share the credit of this work with my lab members, especially Eyal Baz, who is an excellent researcher. And I would like to thank my supervisor, Professor Abdussalam Azem, for his support throughout the project.
So back in the day, you did your bachelor's from JP University for biotechnology from Himachal Pradesh in India. And then you took up a job at an IT company, Infosys. I think it's a leading company in IT known across the globe.
Yeah.
And then you transitioned back to the world of sciences. Would you want to talk more about the transitioning and this whole change that happened in your life and why such a big step?
Yeah, sure. So during my tenure at the IT company, I was very much enjoying my work, but I also sorely missed my natural affinity towards biology. And therefore, to take the best of both the worlds, I decided to pursue a PhD in bioinformatics, specifically in computational immunology, which is essentially the interpretation of immunological data with the help of computational tools.
Wow.
Yeah, so I was fortunate to clear the PhD entrance exam and also to fulfill all the criteria required to pursue PhD in the institute of my choice, that is Soefar University in India.
And that's in Punjab?
That's in Punjab, yeah, exactly.
And how was your time there?
It was amazing. So first of all, I would like to express my gratitude towards Professor Manoj Baranwal, who was my PhD supervisor, and I would also like to thank Professor Svetlana Khaibulina from Kazan Federal University in Russia for teaching me the nuances of review and research article writing.
So it seems that he has a lot of exposure globally. You have worked with the Russians, now you're working with the Israelis. Can you tell us more about your PhD life, your PhD career?
Yeah, sure. So my project in my PhD was based on identification of peptide vaccine candidates against Ebola viruses. So for that, I employed a very stringent computational approach, which catered to multiple algorithms and parameters, and we identified 6 potent vaccine candidates, and further in vitro verification was done, and 2 out of these 6 candidates were proposed for in vivo validation for future applications.
Interesting. And was that something that you really wanted to do? How passionate were you about this?
Oh yes, I was very passionate, and this passion kept on growing as the years passed, and I realized that I took the right decision to join the PhD and pursue it in computational immunology, and here I am.
At Tel Aviv University. Thank you for enlightening me on so many different things that you have done. I see that you completed your PhD during the COVID time. Can you tell me more about the things that were around during that time? It was a difficult time for everyone, right?
Yes, sure. It was a very difficult time for everyone, and it was a time when all the scientists in the scientific world were looking for a cure to COVID. Meanwhile, I joined as an assistant professor at Chandigarh University, and I collaborated with Department of Computer and Department of Biotechnology at Thapar University in the bid to find an answer to COVID. And also, during my brief stint at Chandigarh University, I conceptualized a project to identify a globally effective cross-reactive hepatitis C vaccine candidate.
And can you tell me more about the C project that you were working on?
Yeah, sure. So on this project, we studied all the genotypes of the virus, and we proposed a probable peptide vaccine candidate, which is expected to activate T cells as well as B cells without any side effects. And we also tried our best to ensure its global applicability via HLA and protein peptide interaction analysis. And this work was published in collaboration with Dr. Amit Kesar and David Byrne from Department of Biochemistry at Tel Aviv University, and with the assistance of Claudel, who is a very enthusiastic researcher from Montreal University in Canada.
Oh, you're very, very internationally connected, huh?
In this field, we have to form collaborations, we have to network so that together we can bring a change in the society.
That's such a wonderful job to do.
Thank you.
Very interesting. Truly inspired. So, what brought you to Tel Aviv? What brought you to Tel Aviv University? And what is the plan ahead?
Okay, so during my brief stint at Chandigarh University, I realized that it's very important to have a multifaceted skill set so as to be competitive in this field. And therefore, I wanted to gain intensive wet lab experience to complement my computational skills. And this is where Professor Moshni Ghosh, who was the head of department during my PhD at Thapar University, introduced me to the remarkable work being carried out at Tel Aviv University. And considering my profile, I applied at Professor Azem's lab. And after a few tough rounds of interviews, I was fortunate enough to be selected. And I'm grateful for this opportunity.
Very nice.
And in the future, I would like to continue in academics to fulfill my two desires. That is, one is teaching and one is conducting research. But as I say never. So I'm keeping my options open for industry as well.
That's interesting. That sounds fun.
Also, I would like to inform you that the Faculty of Life Sciences at Tel Aviv University has a collaboration with the Department of Biotechnology at Thapar University in India. And there is a regular exchange of master's program students and bachelor program students. And also now there is a collaboration for the food security projects.
There are other faculties also in Tel Aviv University which are not just working with Thapar, but they're also working with like IITs in India. It's an amazing thing, an amazing collaboration. You're here and you're working with the leading universities across the globe, in India, in the US, in Canada, in Russia. It's an exciting place to be.
Yes, definitely.